All right. Well, hello, everybody. Welcome to PAME's webinar on emerging excellence in epilepsy mortality research. These are highlighting four posters from last year's PAME meeting. We had over 100 poster abstracts submitted, and we had over 50 presenters. And these are sort of the four that we wanted to highlight. And so we have fantastic speakers that agreed to do that for us today. Thanks for joining us. I'm Gordon Buchanan. I'm a professor of neurology. I see adult epilepsy patients at the University of Iowa. I'm also the director of the MD PhD program, and I'll be moderator for this afternoon. And we'd love to see where everybody's joining us from. So if you can share that in the chat, that'd be fun for us to just kind of see our representation. So let's see, Allison, if we can go to the next slide. All right. I just quickly wanted to mention that our today's webinar was sponsored by Neuralis, and we're really grateful to them for the sponsorship. So just some quick housekeeping stuff. Please share questions in the question box. Help the moderator help us keep track of questions. Use the chat to connect with other attendees. This is kind of a fun opportunity for our community to get together. Any questions in the Q&A section, you can kind of upvote questions that you have the most interest in. And there will be a recording of today's discussion available about 24 hours after this. Okay, I think I got. Okay, and just a quick reminder, we're not able to give specific medical advice, so please keep questions kind of general in nature or focused on the science. Yep, okay, great. And again, I mentioned we have sponsorship from Neuralis, so thank you to them for sponsoring this. And I'm just going to quickly introduce our four speakers, and then we'll have them each speak, and then at the end they'll each do like a 10-minute presentation, and then at the end we'll do a panel kind of Q&A. Okay, our first speaker is Dr. Selena Garcia DuBar. She's a postdoctoral research associate in the Wanker Lab at the University of Virginia, where she investigates neural mechanisms underlying epilepsy. She earned her PhD in pharmacology and physiology from Georgetown, where she utilized patch clamp electrophysiology to study neuronal circuitry. In her postdoctoral work, she employs in vivo physiological recording, including electrical stimulation, EEG, EKG, and plethysmography to examine seizure-induced apnea and mechanisms contributing to SUDEP. Our next speaker will be Dr. Eduardo Bravo. He's a Chilean biochemist working as a research assistant professor in Dr. George Richardson's lab at the University of Iowa. His research focuses on how deficiency of the serotonergic neurotransmission can increase the risk of sudden death in infants and in patients with epilepsy, so SIDS and SUDEP. In particular, he is interested in how breathing modulation can help reduce mortality, preventing apneas, and activating the autoresuscitation mechanisms. All right, and then next we'll have Dr. Heather MacLeod. She's a genetic counselor and the senior project manager for the Data Coordinating Center for the Sudden Unexpected Infant Death and Sudden Death in Young Case Registry, where she's been helping to develop and implement the registry since 2013. She's an active member and participant in many professional societies, including the National Society of Genetic Counselors, National Association of Medical Examiners, Heart Rhythm Society, and Sudden Arrhythmia Death Syndromes Foundation. And last but not least, we'll have Anas Reda, who's an MD PhD student at Vanderbilt University working with Dr. Dario Engelhardt. His work focuses on mechanisms underlying epilepsy and sudden unexpected death in epilepsy, or SUDEP, leveraging advanced neuroimaging and intracranial electrophysiology techniques. He's passionate about translating systems-level neuroscience findings into improved outcomes for patients with drug-resistant epilepsy. So welcome to all of you. We're really excited to have you here. So let's get the presentation started. And first, we'll start with Selena.
Okay, let me. Oh no, this is not. Sorry, just a second. So you can see the green.
We see Zoom.
Okay. Um, okay. All right. Does this look okay?
Yeah, looks good.
Everyone's good. All right. So hi, I'm Selena from the Winker Lab at UVA. Before we start, I just wanted to make a quick content note that this presentation includes discussion of seizure-related death in mice. I just want to acknowledge that it can be a sensitive topic, and I really appreciate everyone for being here today. Okay, so this is the one audience that I don't need to sell you on the importance of SUDEP and researching it and why we research it. But I do want to highlight that presently there's no way to really prevent SUDEP. The only thing you can do is prevent the number of seizures, which is an issue for the about third of patients who are refractory to treatment. Um, the pathophysiological mechanisms underlying SUDEP are still not really understood, but it's believed to be a failure of respiratory and cardiac function. So this is why to study SUDEP in a replicable, mechanistically meaningful way, preclinical studies with animal models where death occurs immediately after convulsive seizures are used. And in this presentation, I'm going to use the term SUDEP to refer to both the clinical diagnosis and death after a convulsive seizure in animal models, excluding status epilepticus. Oh no, minimize this. Okay, sorry about that. Okay, so in the rare cases where SUDEP has been monitored in epilepsy monitoring units, such as the Mortimer study, respiratory arrest is secondary to seizures is the primary cause of SUDEP. And as you can see here, terminal apnea starts minutes before terminal asystole. Our lab has previously shown that genetic and drug-induced epilepsy models have demonstrated seizure-induced apnea routinely occurs during the tonic phase here, and that um, there's okay. And if breathing does not recover immediately post-ictally to death, or immediately post-ictally, death is imminent. So here we can see the only difference. Both of these mice have seizures, they have a tonic phase, and then green mouse recovers, the red mouse does not. And we also know that seizure-induced apnea is not impacted by forebrain inhibition in genetic models. So the hypothesis of our lab currently is that overactive brainstem neural circuitry is what produces seizure-induced apnea. So the preclinical studies I just mentioned were carried out in mouse models of monogenic epilepsy, but SUDEP's been documented across the spectrum of epilepsy syndromes. You all know to address this, our lab has developed an aggressive Kindling protocol as a non-genetic SUDEP model of temporal lobe epilepsy, which is the most prevalent form of focal onset epilepsy in adults and can become refractory to treatment. So to implement this protocol, we do stereotaxic surgery in both male and female mice to implant EEG and ECG electrodes, along with a bipolar stimulating electrode into the hippocampus. Mice are given at least 1 week to recover from surgery. And then before the first day of kindling, the stimulating threshold is determined. And then we can begin our rapid kindling protocol after this, where mice are stimulated with 10-second trains of 1 millisecond, 5 phasic pulses at 50 hertz across the hippocampal electrode. This is repeated 10 times a day with 20 minutes in between for 3 days. And this is a schematic of a mouse undergoing the protocol. It has the headset, the stimulating electrode here, EEG, ECG leads. It's connected to the stimulation isolation unit. The pressure transducers here, which is how we can measure the breathing. Everything goes into amplifiers and then is digitized so we can view it on the computer. And all of this is videotaped so we can review the footage afterwards as well. Okay. So using this model, we've been able to generate seizures without apnea and with apnea in both fatal and non-fatal seizures. We've scored seizure behavior with our modifying racing scale from a 0, which is where we see ictal activity but no seizure behavior, all the way to seizure-induced death after a tonic seizure with apnea. So this is an example of a stage 6 seizure where we observe wild running and jumping right here. And it most closely resembles severe clonic seizures. This is an example or representative trace of a stage 7 tonic seizure that's not fatal. And you can see it has this tonic phase here, which you can see in the ECG. There's apnea and then hind limb extension here. You can see here. And then our stage 8 describes seizures that meet all the qualifications for a stage 7, but they immediately lead to death. So the seizures that we produce in our kindling model are nearly identical to the spontaneous seizures we've observed in the past in our lab with genetic models of epilepsy. Okay, so using our rapid kindling model on in C57 black 6J wild type mice, this is a heat map of each stimulated seizure over 3 days, with each row representing 1 mouse and each box representing 1 seizure. So you can see that seizures stage 5 or below, which are less severe clonic seizures and non-clonic seizures, are in shades of blue. The severe clonic stage 6 seizures are in a maroonish dark purple color. The 7s are in a lighter purple color, and then the 8 is in red. And so the seizure that they have that leads to death has a little white cross on them. So in the wild type mice that we've kindled, about 33% of the mice died from seizure-induced apnea. And the survival curve shows the decreased probability of survival as a percentage with repeated hippocampal stimulations. Okay. This figure shows the onset of apnea in blue for each mouse, and then terminal asystole in red. And as you can see, all of the mice stopped breathing minutes before their heart stopped, which is similar to the Mortimer study patient data in humans. So 100% of the fatal seizures were tonic seizures, so no fatal seizures came after a clonic seizure. And here you can see the respiratory frequency and heart rate over time, with 0 adjusted to apnea onset here, with the yellow box representing the time range of stimulations. So respiratory frequency for mice that had non-fatal stage 7 seizures are in blue, and fatal are in black here. As you can see, it's similar to the image I showed in the beginning of my presentation from genetic models of epilepsy. So it's consistent across models. This is similar, but the heart rate data, and you can see both groups show decrease in heart rate after seizure induction, so after the stimulations. This is where apnea starts here. Then you can see that the stage 7s recover, whereas the stage 8s continued in a downward trend until reaching 0. So this is an example of a typical severe chronic stage 6 seizure, like I showed before. But interestingly, while 100% of the tonic seizures have an apnea, even if they're not fatal, in going through the data, we observed apnea in a subset of stage 6 seizures that did not correspond with the tonic phase, which was reconfirmed with our video recordings we have. This is a histogram that shows the frequency distribution of the longest time between breaths. Stage 7 non-fatal seizures are shown in blue. Aren't as many of these. For all seizures scored as 6 based on the wild jumping and running behavior, about 26% of them had a pause in breathing greater than 1.5 seconds, which is what we were using as our cutoff for apnea. Instead of adding an additional stage to our scale, we refer to these seizures as stage 6 seizures with apnea because the behavior looks the same. They just have an apnea versus not having an apnea. This plot shows that tonic seizures have significantly longer apneas on average than the stage 6 seizures with apnea, with a Poisson mixed effects model. These bottom graphs are similar to the ones I showed before, and the tonic phase in blue is the same as I showed before, just with the stage 6s shown here. So the stage 6 with apnea is in green, and then stage 6 without apnea is in orange. And as you can see pretty clearly, the shapes of the seizure types with apnea are more similar than the ones without, which is more of a straight line instead of this dip when the apnea occurs. So in the earlier slide, I discussed our previous finding that seizure-induced apnea is not impacted by forebrain inhibition, which has led us to utilize our Kindling model to examine neurons activated during seizure-induced apnea in the brainstem. So using the same rapid Kindling protocol we used for our wild-type mice, we use transgenic TRAP2 mice, and we kindle them until reaching their first stage 6 or stage 7 behavioral seizure. So this is a heat map of these mice, and we continue past 3 days to try to get more of the different seizure types here. After we get their first stage 6 or 7, we inject them with 4-hydroxytamoxifen, which is right here, and then we wait about at least 2 weeks for TD tomato expression and perfuse and collect brains at this point. So here, after the TD tomato has had long enough to express and we perfuse them, we section the brains to examine the TD tomato fluorescence, and we found more activated neurons in the PAG of mice that had seizures with apnea versus seizures without. So here's the PAG. This is a stage 7, so the stage 7s always have apnea. This is a stage 6 that didn't have apnea, and then this is a stage 0, so a mouse that had a seizure with ictal activity but no seizure behavior. And then this is just showing in comparison to the colliculus and motor cortex. In a subset of these experiments, we also performed RNA-scope or in situ hybridization to assess the subtypes of these activated neurons observed. And we observed the majority of the activated cells to be co-labeled for BGO2. So this is the co-labeling with BGO2 versus GOT1. So majority of the activated cells are glutamatergic. Okay, so in conclusion, our model reliably produces seizure-induced apnea and SUDEP with 3 days of kindling and can be used to further investigate differences between seizures with and without apnea. Histology suggests excitatory neurons in the perioccipital gray are activated during seizures to produce the apnea that leads to SUDEP, and the PAG represents a potential target for intervention to prevent seizure-induced apnea. SUDEP. Okay, and thank you to everyone in the Winker lab and at UVA. If you want to learn more about our lab or our research, feel free to email me. And thank you, everyone. Yeah, stop sharing.
Great. Thank you, Selena. All right, that's great work. Next up, we have Eduardo, so feel free to share. Testing everybody's technological savvy.
Give me one sec here. Share the screen. Yep. So sharing the right one. Can you see it over there?
We see your
Yes. No, well, that's my hometown. I need to show off a little bit.
all right.
Okay, so thank you everybody to be here assistant. Thank to the organizer for this invitation to participate on this session. The poster that I will present is called Increased Risk of Fatal Apnea when the amygdala is stimulated during loss of wakefulness. But before getting deep into the poster, is like, why the amygdala? Where the amygdala is part of the limbic system, which one is the major process center for emotions, such as fear, anxiety attack, panic attack, and hyperventilation. All of them can modulate breathing. Actually, in 2024, Travis and Bao studied how all this emotion can modify breathing, and he drew this all the possible input-output from the central amygdala to different nuclei involved in the breathing modulation. But the story between seizures and apneas doesn't start so late. It started really early in 1899 with the first anecdotal notes that the patient turn blue when they have seizures. Later on in 1968, Nelson and Ray described that patient during seizure stopped to having chest movement, so assuming that they were apneas. But was not until like 1972 when the first experiment in monkey by Bombalet and Bobo was run, and they stimulate different nuclei of the amygdala and surrounding nuclei and see how the breathing is modulated. So this one is one of the figures of that paper, and you can see different breathing traces related with which nuclei they were stimulating. The black part below the breathing trace is showing up the period of stimulation, and I want you to focus over here. That is the central nucleus of the amygdala stimulated, and you can see that induced a profound hypoventilation. But to move fast forward for today's, it's in 2015, Brian DeLuey showed that when the amygdala propagate into when the seizure propagate into the amygdala, that induce apnea. And if we stimulate the same area of the amygdala, apnea can be induced, no seizure related. That work was expanded by William Novice and Nuria Lacquey in 2018 and 2019, respectively. And they expanded two regions from the hippocampus and amygdala stimulation inducing apneas. And Ariane Rohn, that is part of the Brian DeLuis group, working with pediatric patients, she showed like when they stimulate a specific area of the central amygdala, they can induce apneas. This work is really remarkable in the way that they're using a really tiny electrode since they're pediatric patients. So the space between the contact is really small. So that gives it to them a really tuning tool to mapping the amygdala and represented in a 3D ways. So there you can see in gray what is the amygdala area, and in pink, which one is the specific area of the amygdala that when they stimulate it, that can induce apnea. They define this area as airside, or amygdala inhibition of respiration. Why this is relevant? As Selena mentioned, in the Mortimer's study, they show that the leading cause of death is apneas, then they develop bradycardia, and at the end, terminal asystole. And I will say thanks to Selena for saving me a lot of time, because she represents and she expressed that we can recapitulate those phenomena in mouse, different mouse model. Of epilepsy mouse model. In this case, I'm showing like an Dravet syndrome mouse model, in which one you can see in the first trace is the EEG, so you can see the seizure, then the power spectrum analysis of the seizure, The second trace is the EKG, and you can see that the EKG persists even for a minute after the seizure is ended. Remarkable breathing, that is the last trace, stop immediately when the seizure is ended, and they never recover again. So if we are able to recapitulate all the pathophysiology of SUDEP, how important or is the amygdala playing the same role in mouse than in human? So I take the task to map the whole amygdala in a 3D way. So that is on a 2 millimeter long section using head-out plethysmography system. That allowed me to combine the plethysmograph with stereotax and being able to moving the electrode around the amygdala, stimulate it and recording in vivo which one is the response on breathing, if I'm inducing apnea or any breathing changes. So when I compile all the information together and try to represent it in a diagram, here you have a sagittal slice of one of the hemisphere of the brain, a mouse brain, and it's highlighted in the square the different distribution of subnucleus of the amygdala. Why that is relevant is because I will overlapping all my information becoming from breathing into that square section. What you can see over here is the heat map of breathing modification. In dark blue is the area that I am able to induce total apnea, so it's no breathing at all. In green is when the breath is not changed or slightly changed, and in yellow is areas that if I stimulate it, I induce an increase in breathing frequency. Each one of the squares from the left to the right are indicated like in a medial or more lateral section in this sagittal slice. And each square from left to right is from rostral to caudal. And you can see over there that is from 0 bregma up to 2.5 bregma, minus 2.5, and from dorsal to ventral. So it's in a really tiny area that I am able to stimulate and induce apnea. And if I overlap that blue dots in the diagram, that is the area that I can just, that I need to stimulate to induce the apnea. All the other ones have no effect or even increasing breathing frequency. But how powerful is this modulation of breathing if I stimulate the amygdala? Well, it's really powerful if I'm using light anesthetized mice. The first graph is a baseline of this mouse that is slightly anesthetized. For that reason, the breathing frequency is kind of low. But the important message here is I can induce apnea as long as the stimulus is persist. So I can induce 10-second, 40-second, 60-second, even 4-minute apneas on those mice. Unlucky, I'm not able to replicate these profound apneas on free-running mouse. Why? That is a big question for us. Is like because basically on patients, apnea has been inducing during awake. One of the possible explanation is species difference, but that is way broad. What makes the difference between these two, between mouse and human, beside the size? It's how you collect information from the environment. Human is more visible and hearing. Mouse are vision, but also is really powerful, the sniffing. So the wakefulness drive of breathing in mouse have a higher hierarchy on them than in humans. The other difference is like on patient, they can be distracted during the trial, and I still do not found the way how distract a mouse before the amygdala stimulation. So if any have any idea, I'm open to receive your suggestions. This one is how I can mimic this lack of wakefulness then. Well, I have two options. One option is seizure related. When seizure spread or seizure hit the brain, I have a decrease in cortical activity. With that one, I will have a depletion of the wakefulness drive of breathing and volitional control of breathing. But I want to study the network and the mechanism, so I need to take it out the seizure away. What is my second option to try to run the same lack of wakefulness drive? That is stimulating the amygdala during sleep. During a sleep, the wakefulness drive of breathing shut down and the arousal system take over the breathing, the automatic breathing. So for that, thanks to a PhD student here in the lab that she developed this code in Python, we're able to combine the analysis of EEG, EMG, and plethysmography all together to a score and create like an hypnogram or somnogram or sleep-awake architecture in a really accurate way. Actually, at the left, you can see the road traces. At the right, you can see the power spectrum analysis. Together with the purple line is the delta theta ratio. And in the bottom is power spectrum of plethysmograph. And you can see how highly correlate the power spectrum of plethysmograph is according to the sleep-awake architecture. The sleepnogram is shown in red, awake, in blue, no REM, and in green, REM. So now that I have this tool, I can stimulate those mice during awake or sleep time. In awake, as I told you, I was unable to induce this apnea. Actually, with a stimulus of 3 seconds with 50 hertz, I'm able to disrupt breathing or increase breathing frequency, but not much inducing apneas. Using electrical stimulation or optostimulation is the same. When I quantify those data, is just 8% of the time I can induce the apnea. So 92% of the time, breathing doesn't change or increase breathing frequency. In the other hand, if I'm stimulating during sleep, the story change. It's the same stimulus, 3 seconds, 50 hertz, and I can induce a complete apnea that is prolonged even after the stimulus is ended. In the power spectrum analysis, you can see that the breathing is completely abolished even for 15 seconds after the stimulus end. And when I quantify those data, it's like 64% of the time now, not 8, I can induce those apneas. And most relevant is in 9% of the time, these apneas become fatal. So the mouse is unable to back and breathe again. An interesting point here, after Chiara developed this tool, is like I can figure it out that 90% of those 9% of the cases, when the apnea becomes fatal, is when the stimulation was performed during no REM. And in the last PAME session, we saw like a bunch of data related with how deep or how increased fatality could be if the seizure hit during no REM. So wrapping up all the information led me to the conclusion that amygdala play a role, an important role in modulation of breathing. Stimulation of the rostral central amygdala can induce prolonged central apnea in lightly anesthetized mice and in lack of wakefulness drive. That this phenomenon may contribute to the SUDEP during sleep, exactly for the same reason, lack of wakefulness drive and lack of chemoreceptor drive of breathing. And the propagation of the seizure into the amygdala can increase the risk of fatal apneas. So before the end, I need to just say thanks to everybody that has been collaborated in a tutor way, help me with experiment or planning experiment or just try to explain data. So thank you very much.
Awesome. Thanks, Eduardo. All right, and next we'll have Heather.
Hello. Let's see. Can you see my slides?
Yep, you're not in presentation mode yet, but yeah.
Awesome. Here we go.
There you go.
Jordan, and thank you to PAME for allowing us to present this important data again. I work on the sudden unexpected death and sudden death in the young case registry, and this is a review of 10 years of our data. The SUID and SDY case registry is funded by the US CDC and NIH, and these contents are solely the responsibility of myself and should not be construed as the official position or policy of the government. So, why study sudden death in young people? It is a public health concern. It's a critical issue requiring comprehensive investigation and systematic data collection to understand the causes and risk factors. The SUID and SDY case registry is a surveillance study designed to collect detailed information on sudden deaths in young people ages 0 to 20 years old. Data is stored in the Pediatric National Fatality Review Case Reporting System that I'll refer to as the CRS. This is the structure of the case registry. It has both a surveillance component, meaning that we look at all of the young sudden deaths in a funded jurisdiction. So we know both the numerator and the denominator. We know how many children live in the funded jurisdiction. So this is not a national registry. It is funded in only certain states and jurisdictions, as I will show you a map in a moment. And the purpose is to create a registry of information about each case, including death certificates, medical records, death investigations, autopsy reports. And in the development of the registry, we also included to collect a blood sample at the time of autopsy so that families can consent to either DNA banking or participation in genomic research. The research component, we are making the genomic data from the cases where the family consented available to researchers to better understand the mechanistic, genetic, and other causes of sudden death in young people, including SUDEP. So this is a schematic of the multiple government agencies involved in funding the registry, the CDC, and National Heart, Lung, and Blood, and National Institutes of Neurological Disorders. This is a map of the places where the registry is active. As I mentioned, our data goes to the CRS, which is housed at Michigan Public Health Institute, and our biospecimens go from the medical examiner and coroner offices to the University of Michigan, where they're extracted for DNA. We then make these data sources available to researchers. This is a current map of where the registry is active. So we have some sites that are SUID only, so only sudden unexpected infant death, and those are in dark blue. And then in light blue, we have the areas that do both the infants and the older children. Our process for doing better surveillance on sudden death in young people integrates multiple data sources, including demographic data, medical history, autopsy, and pathology findings. And then on the right here, you'll see we follow a standardized algorithm to incorporate the findings of each case. And then we assign a specific category to each case. And this is what helps us determine incidence. Like I mentioned, we know both how many children die in each jurisdiction and how many live there. And then once we assign a category, we can calculate incidence. How often is our sudden deaths in epilepsy happening? The process at the state or jurisdictional level looks like this. A sudden death in a child occurs, an autopsy is performed, and samples are saved. And then the cases go through multiple levels of review, both at the level of child death review, as well as an advanced review, which has more specialized medical professionals, including pediatric cardiology, genetics, and epileptology. The data is then analyzed and disseminated to help inform prevention, and the data and biospecimens are used for research to better understand the causes of sudden unexpected infant death and sudden death in young people. So these were our methods. Our data source was the CRS, where all our data is stored. We looked at deaths from the beginning of the registry, which began collecting data in 2015 through all our current cases in 2025 at the time this analysis was done, which was last summer. We used cases that met our case definition of sudden and unexpected and were in participating jurisdictions. Over time, this is the third funding cycle of the registry, and currently 12 sites are funded for SUID and SDY. At one point, we were up to 13 jurisdictions. So we included the data from all those places. All cases were QA complete, and all cases were categorized using the case registry algorithm. And we also looked at, there are definitions by NASHF to better understand sudden unexpected death and epilepsy. So I will present both sets. So from our algorithm, we looked at the unexplained sudden unexpected death in a child with epilepsy and seizure disorder cases, and the unexplained possible cardiac and sudden unexpected death in a child with epilepsy seizure disorder. We have a category where it combines, like, perhaps they had a diagnosis of epilepsy, but maybe they also had a heart condition. And so we capture those deaths in a combined category. For our purposes, even though drownings and motor vehicle accidents are not included in the NASHF definitions of SUDEP, in the registry, we do include them. So we also looked at those in this analysis. So we started with 6,400 case registry cases that were QA complete. 278 of the cases met criteria for this analysis of either being sudden unexplained death and epilepsy or the combined category of SUDEP and possible cardiac. The breakdown was 244 cases were SUDEP and 34 of our cases were SUDEP/possible cardiac. So importantly, of all young, sudden, unexpected deaths in the registry, this accounts for 4% of cases. We found that compared to other registry cases, the autopsy rate was lower. Only 79% of our epilepsy cases had an autopsy conducted, which is much lower than the overall registry, which is 92% have a complete autopsy. We found that 18% of our cases had a known genetic diagnosis prior to death. Dravet was the most common. When we applied the NASHF definitions, we found that with our cases, 60% met the definition of definite SUDEP. 43% had abnormal brain findings. Sorry, I think that was meant to be a sub-bullet. 7% met the NASHF definition of definite SUDEP plus, and this was based on either cardiac findings or significant family history. 16% were probable SUDEP plus because they had no autopsy. 4% were possible SUDEP, again with no autopsy, and 13% of our cases were determined to be not SUDEP, and that was largely the motor vehicle crashes and drownings. But at our advanced review teams, they found that in those cases, they thought that epilepsy had played a role. In conclusion, the lower autopsy rate in our epilepsy cases, we believe may reflect the complex medical history of some of the children, the assumption death was epilepsy related or due to another chronic condition, as well as possibly the forensic workforce shortage. There is a huge shortage of forensic pathologists. I think they are working at about 50% of the level they need to be at, and that could be contributing to these cases not getting a full autopsy. Increasing autopsy rates could identify more cases that would qualify as definite SUDEP. And 2/3 of our cases met NASHF criteria for definite SUDEP or SUDEP plus. And to us, this suggests that on a public health level, simpler definitions may be sufficient. I'd like to thank our whole team. It definitely takes a village to do this work. In particular, the states and jurisdictions that share their data with us. Thank you.
Great. Fantastic. Thanks, Heather. That was great. All right. And then last but not least, we have Anas.
Awesome. Can you guys hear me and see my screen?
Yep, looks good.
Perfect. All right. So, hello, everyone. My name is Anas Reda. I am an MD PhD student at Vanderbilt University under the supervision of Dr. Dario Englot. Thank you so much, Dr. Buchanan, for the invitation, and thanks to you too, Allison. So today I'm going to talk about very briefly about impaired arousal circuitry in epilepsy, some neural substrates of respiratory vulnerability, and specifically talk about this brain region called the BNST, or the bed nucleus of the stria terminalis, as a potential target for neuromodulation in patients at increased risk for SUDEP. So I'll just begin by very briefly talking about SUDEP. And like everyone else before me mentioned, SUDEP is truly the single most devastating consequence of epilepsy. In patients with drug-resistant epilepsy, it likely accounts for about half the deaths. Sadly, we really don't understand SUDEP. And I think, this is exemplified by this paper from 2020. So back in 2020, when this paper came out, really the two biggest risk factors for SUDEP were, one, having a tonic-clonic seizure in the last year, and two, living on your own. You can see that, and then together, these kind of combine to increase your likelihood of SUDEP by 67 times. You can sort of understand why these are risk factors, but they are not biomarkers, and they're not things we can look for in the brain. But very recently, literally within the last year, two really awesome papers came out, and they really showed this progression into SUDEP. And the idea is that when patients have seizures that cause ictal apnea, so they get apnea or cessation of breathing during the seizure, that's super dangerous. When the seizure ends, it turns out that for some of those patients, they actually keep being apneic. So even though their seizure ends, they keep having episodes of apnea. And we think it's these repeated episodes of apnea in succession that eventually results in SUDEP. So kind of to put everything together, in an epileptic brain, we think that repeated ictal insults, which is just a fancy way of saying more seizures, these seizures, they damage very specific brain regions, making the patients have poor CO2 chemoreception, which is just a fancy way of saying when your oxygen goes down, patients with this damage can't detect it. When your oxygen goes down and you can't detect it, your brain can't wake you up, can't arouse you. And perhaps that's when patients succumb to SUDEP. So kind of with this understanding, what brain regions should we look at if we're interested in sudden unexpected death in epilepsy? Well, a great place to look is the basal forebrain. Why is the basal forebrain important? It's because it's strategically positioned between brainstem arousal and respiratory nuclei and cortical arousal networks. And we think that when your brainstem is sensing the levels of CO2 and oxygen in your blood, that it kind of talks with the BNST, and the BNST tells your cortical networks to wake you up. So it literally sits in a very privileged anatomic position between brainstem respiratory nuclei and cortical arousal networks. Second, we know that within the basal forebrain, which is technically this whole part, sits the BNST, or the bed nucleus of the stria terminalis. This is a very important brain region that is thought to be an integrative brain region. One of the things it integrates is chemosensitive information. So it itself is not chemosensitive, but it receives a lot of information from intrinsically chemosensitive neuronal populations to alter its activity. We also know that the BNST is technically part of the extended amygdala. And as Dr. Bravo said earlier, we know that stimulating the amygdala in very specific places can both cause apnea while stimulating and even persistent apnea after the cessation of stimulation. And then lastly, as was mentioned also, we know that in a mouse model of Dravet syndrome, which is an epileptic encephalopathy at increased risk for SUDEP, we see that the neurons sitting in the PNST and specifically connecting to the parabrachial complex are hypo-excitable. So they're not working super well. So we kind of take these things and are like, okay, this brain region sits in its very specific anatomical region. We know its normal job is probably to relay this chemosensory information with arousal, right? And we know that in a mouse model of Dravet syndrome, it doesn't seem to be working well. So can we go and study it in patients? So when we want to study in patients, really the easiest way to do this and most straightforward is to just use fMRI. And I'm not really going to belabor the details of fMRI, but I'm going to try to talk about what fMRI tells us. So fMRI tells us how well two brain regions talk to each other. And it can tell us how they talk to each other kind of just generally. And we call that non-directed functional connectivity. So if their functional connectivity are high, they talk to each other well. But then in a conversation between two brain regions, yes, they're talking to each other, but it's also important to know the direction of the conversation, right? Is brain region A talking to B, or is B talking to A more, et cetera, et cetera. So I'm not going to go into these details, but ultimately, we just took 33 healthy controls, 37 patients with temporal lobe epilepsy specifically, and tried to understand the function of the bed nucleus of the stria terminalis, or the BNST, using these connectivity measures. So the first thing we always ask when we're studying a brain region is, hey, how does this brain region connect to the rest of the brain? And when we do that, right, we find that compared to our healthy controls, which are in this blue, we find that patients with epilepsy have significantly lower functional connectivity between the BNST, that is ipsilateral to the seizure focus, but that doesn't really matter, to the rest of the brain. And then when we look at the functional connectivity in a directed manner, we find the same exact idea. And the idea here is that not only does the BNST stop talking to the rest of the brain, but this loss of communication is specifically in the BNST to the rest of the brain. So the BNST stops talking to the rest of the brain. It's not that the rest of the brain stops talking to the BNST. And then we can do some fancy analyses called voxel-wise analyses. All this is showing us is that in these blue areas, we find that in patients relative to healthy controls, the BNST has worse connectivity to these temporal and subcortical regions. So kind of put together, we show that the BNST has poor connectivity to the rest of the brain in patients relative to healthy controls. But that's kind of looking at the whole brain, right? The whole brain doesn't really do the same thing. So let's take the brain, break it up into all these cool separate networks, right? We know in the brain we have a visual network, a language network, a this network, and that network. So let's take the brain and break it up. And we broke it up into these different networks, which are the default mode, dorsal attention, all of these. The one to really, really pay attention to is this ventral attention network. The ventral attention network is super important because it is a bottom-up alerting system. So in the case of a patient with epilepsy undergoing some sort of respiratory challenge, right, meaning their CO2 is increasing, this is the system that we think that's going to tell the patient, tell the body and the brain, hey, wake up, the CO2 is increasing. So in patients with epilepsy, we find decreased connectivity of to the ventral attention network, And the easiest way to think about this is that your alarm system, which tells you, hey, wake up, wake up, wake up, isn't working anymore. When we look at the opposite direction, we find that the dorsal attention network actually increases its connectivity to the BNST in patients. We think that this is kind of a compensatory things that happen. So in the ascending direction, patients lose connectivity from the BNST to this alarm system network. When you lose your alarm system, maybe your brain is trying to tell the BNST in the reverse direction, hey, I'm not getting any information from you, talk to me. Next, instead of looking at the BNST and the cortex, we looked at the BNST and specific brainstem nuclei. Those are the nuclei at the bottom of the brain that are important in arousal and respiration. When we look at the functional connectivity, we see no differences between controls and patients. But then when you look at the directed connectivity, we see a decrease in BNST causal influence onto the VTA, the median RAFA, the cuneiform nuclei in patients relative to controls. And there's a decrease in the amount of information the BNST sends to these. Interestingly, there is no compensation. Remember earlier how I showed you that the connectivity decreases from the BNST to the ventral attention network, and then we think there's a compensatory dorsal attention network telling the BNST wake up. Here we don't see that compensation. So from BNST to these brainstem nuclei, the connectivity decreases, and there's really no compensatory increase. So putting all of this together, why do we think that the BNST is important? In patients without epilepsy, right, their brainstem nuclei are sampling their blood. They're saying, okay, this blood has enough oxygen and it doesn't have a lot of CO2. I'm good, I'm happy. But when the CO2 goes up a little bit, these nuclei, they talk to each other, they talk to the BNST and they tell it, hey, tell the human to breathe more deeply or kind of breathe better. In patients with epilepsy, they have something called a seizure onset zone or an SOZ. When the SOZ discharges all that electricity, all that epileptic activity, it travels to the BNST. And we know through kind of decades of experiments that when seizure activity travels to these brain regions, it messes up their function. So the idea here is that in a patient, especially with focal to bilateral tonic-clonic seizures, the spread of seizure activity destroys this arousal mechanism, resulting in them succumbing to respiratory challenges. So putting everything together, we find that patients with epilepsy have poor integrative role of the BNST. We also find specifically that the BNST loses causal influence on the ventral attention network. Again, that alarm system that tells healthy controls that, hey, wake up, too much CO2 in your blood. The same, we find very similar things towards brainstem nuclei. And then lastly, and I think most importantly, if we are saying that, hey, we think this brain region is not arousing patients correctly, can we stick an electrode in it and stimulate it to make it work better? So hopefully that's the answer. And with that, thank you again so much for the invitation. This really, really means so much to me. And of course, none of this would be possible without our phenomenal team and my mentor, Dr. Dario Angela. Thank you so much again.
Great. Thank you, Anas. Thank you to all the speakers. We really appreciate you all talking. This was obviously a bit of it was challenging to do 4. It was a big ask to do 4 in this short time. So we're at time, but we can do probably 1 or 2 questions if people can hang around. It looks like we had some discussion already. I know Eduardo fielded a question in the chat. Any questions from anybody? Anas, here we go. So a question for Eduardo. Understandably, the SUDEP research community is focused on the areas of the amygdala that cause apnea after stimulation. However, that areas of the amygdala that increase breathing when you stimulated them are also interesting. Do you think that stimulating those areas would alleviate seizure-induced apnea and prevent mortality? Have you tried any experiments like that? You're muted.
I figured that I was mute. I think that would be depending on, because all the mapping, I did it with light anesthesia. So basically it's not in a wake related status. So depending on the role of this specific amygdala area, our PAME during a wake, it could become like a possible chance in a closed loop related, if you stimulate, you inhibit the inhibition. So, I mean, it is possible, but unlucky, I don't have the right or the straight answer for Benton.
All right. Anas, I have a question for you. Do you have an idea of where the sensor might be? Like sensing the changes in like CO2?
Yes.
Well, for Anna, go.
Yeah. Dr. Bravo, I think your answer will probably be way more accurate than mine, but I can try. So as I understand it, there are very specific sets of neuronal population, nucleus trapezoidus, STN, for example, whose neurons are intrinsically chemosensitive, which means that their action potential firing rate changes specifically in response to pH. And those are a lot in the brainstem. The question is, can we record from the brainstem? It's very difficult to put electrodes in the brainstem. I know in Canada, they're much more kind of bullish about that, especially, but honestly unsure. So yeah, what's the best way to study brain regions that are intrinsically chemosensitive? Probably EPHIS, but yeah, sticking electrodes in the brainstem will be, I think, a major challenge.
All right. Thank you. Okay, we probably should wrap this up. We're a little over time. I just want to give a quick plug for the PAME meeting, December 2nd and 3rd in Denver, Colorado, the day before, basically the day and a half before AES meeting. You can contact Allison if you need details or check out the website. A lot of you have been there. This is a fantastic meeting that brings together kind of all stakeholders related to seizure-related mortality. Most of us will probably be there. So check it out. Please come submit an abstract, and maybe next year we can get you participating in this webinar. And again, we wanted to just thank Pammy wanted to thank Neurellis again for sponsoring. Really, we can't do this without the sponsorship. So really appreciate that. And again, thank you to Selena, Eduardo, Heather, and Anas. These were fantastic talks, really interesting stuff. And I appreciate everybody being here and participating.
May I say something really tiny, really small?
Yeah.
The whole how important is PAME? The whole idea of this research, how it's going on, was created in 1 year before, so in 2024, in a PAME meeting together with people from other labs. So there you have the time for interact and try to create new path.
And yeah, I see some questions about recordings. There will be recording available. All right. Thanks again, everybody. Enjoy the rest of your afternoon.
Thank you.
Thank you.